We have looked at atomic structure, electrons, electron orbitals, how electrons are involved in forming different types of chemical bonds. That allows atoms to be joined to form molecules. We're ready now to look at the specific molecules that are found in living cells. These molecules can broadly be classified into two groups, the inorganic molecules and organic molecules. The inorganic molecules include water and the various ionic compounds, salts, that are found in living cells. The organic molecules are any molecules that can... Carbon is atomic number 6. If you remember, it has 4 valence electrons, so it would like to have 4 more. So carbon atoms typically form some combination of 4 covalent bonds. That can be 4 single covalent bonds, or they could have 2 double covalent bonds. What we see quite often is carbon forms a linear chain. So you see the diagram here where we have a whole chain of carbon. These are bonded to one another, and then also we have hydrogen atoms that are bonded to carbon. This would be an example of a linear hydrocarbon chain. Remember, the dashes represent single covalent bonds, or one pair of shared electrons. And so if we were to count each of those carbons, they do in fact have 4 single covalent bonds. Carbon can also form a ring structure. So in this diagram, we have a hydrocarbon ring. Again, contains carbon and hydrogen. Now, a question. If you look at these molecules, these hydrocarbons, would they be polar or nonpolar? Remember, we mentioned that typically we're going to see oxygen and nitrogen. Those are the electronegative elements that try to steal electrons away and form polar covalent bonds. These molecules, there is no oxygen, there is no nitrogen. These are very nonpolar. In each case, these bonding electrons are shared equally. So since they are nonpolar, there is no partial charge separation. So would we call these molecules hydrophobic or hydrophilic? Remember, hydro means water, philic means loving. So in the previous segment, we said anything that will dissolve in water is hydrophilic, and that typically would be ions or polar molecules. These hydrocarbons are very nonpolar, so these are very hydrophobic. These will not mix with water. Vegetable oil is long hydrocarbon chains, very, very nonpolar, and as you know, things like oil won't mix with water, very hydrophobic. We can change the activity, the behavior of these, however, if we add some functional groups to this hydrocarbon chain. So there are several functional groups that we need to be familiar with. We need to be able to recognize these. And again, if we add enough of these functional groups to that hydrocarbon chain, perhaps we're going to change the way it behaves in water. The first functional group that we need to recognize is an NH2. This is called an amine group. It is a nitrogen that has 2 single covalent bonds to 2 separate hydrogen atoms. So we'll see some NH2, an amine. We're going to see a lot of hydroxyl groups. That would be an OH. And another one that we will see is called a ketone. This is an oxygen that has a double covalent bond, typically to a carbon. Now, if we look at these functional groups, are these polar or nonpolar? Okay. We see some nitrogen, we see some oxygen. So those are the electronegative elements. Going to steal some electrons away. And so these oxygens and nitrogens, especially if they are bonded to a hydrogen or a carbon, that's going to be a polar covalent bond, and so we have a polarized molecule. We have some charge separation. And so anything that is polar might then be hydrophilic. It might mix with water. Our classes of biological molecules, these are organic biological molecules, are carbohydrates, lipids, proteins, and nucleic acids. So we're going to look at each of these, spend a few minutes looking at each of these classes of molecules. We're going to build some very large molecules, some very large carbohydrates or proteins, lipids, and when we build those, we're going to see the same reactions over and over and over. A reaction that is called a dehydration reaction, or sometimes called a condensation reaction, and this is used to build larger molecules. We're going to take two molecules and we're going to put them together. And so what happens, you see in the top of this diagram a monomer. That would be something like a sugar. If we want to connect two of those together, we can take a hydroxide functional group from one molecule and remove that. Take a hydrogen atom off of the second molecule, put those together, that forms water, and in doing so we can form a new chemical bond that connects two larger molecules together. So you see here in this diagram we have removed water, and in doing so we have connected these molecules together. We're going to see this dehydration reaction over and over and over. Now these are reversible. We can go the other way. So if we are breaking something apart, it's the same reaction going in the other direction. This is called a hydrolysis reaction. So down at the bottom we have a large molecule. We want to break that apart. And so we can break that bond. At the same time, a water molecule is split apart, and we add the hydroxide group to one molecule, and we add the hydrogen to another. So hydrolysis reactions and these dehydration condensation reaction, same reaction going in a different direction. So in one case we are building larger molecules, in the other case we are breaking something down. One way that I can remember this and keep this straight is the term hydrolysis. Hydro means water, lysis means break. And so what's happening in this hydrolysis reaction, we are breaking a water molecule. We are also breaking that large molecule into two smaller components. So we'll see these reactions over and over. Our carbohydrates is our first class of organic molecules. Carbohydrates are sugars and starches. The primary function, biological function for sugars and starches is energy. This is how we get much of our energy, or we store our energy. There are some structural components in cells, especially in plant cells, that are made from carbohydrate. Carbohydrates are composed of carbon and hydrogen and oxygen, and it's always in a 1 to 2 to 1 ratio, so 1 carbon for every 2 hydrogens and 1 oxygen. We're going to see this is a hydrocarbon chain, but we're going to put some functional groups on that hydrocarbon chain. The functional groups that we're going to see will be hydroxyls, a lot of the OH hydroxyl groups, and then there is one ketone group on carbohydrates. A monosaccharide is a single simple sugar. So this would be a carbohydrate, more specifically a monosaccharide, a simple single sugar. Glucose is a monosaccharide, a simple sugar. We also call it a hexose. Hex means 6. And this refers to the fact that glucose has 6 carbon atoms. Here's a diagram of glucose. Now don't let this scare you too badly. If we start over here on the left, you can see the carbons, and they're numbered 1 through 6. Each of those has an OH, a hydroxyl group in it. And there was a ketone. When glucose is dry, it's a linear molecule. Whenever you put glucose in water, it closes up on itself and forms this ring structure. And whenever it forms the ring structure, the ketone, the oxygen of that ketone group gets trapped inside the ring. Again, don't let this frighten you too badly. We see the molecular formula down at the bottom, C6H12O6. Now this would be the formula for glucose, so it's made up of carbon, hydrogen, and oxygen, and it's in that 1:2:1 ratio. So a monosaccharide is just a single sugar, such as glucose. A disaccharide is two monosaccharides that are joined together. Now we're going to join these together with that exact same dehydration synthesis reaction that we saw just a moment ago. So in the diagram over on the left-hand side, we have two glucose molecules. And you can see shaded in blue there, we have a hydroxyl group on one glucose. We're going to take that hydroxyl group along with a hydrogen on the other glucose. Those are going to be removed. That forms a water molecule. And in doing so, we form a new covalent bond between a carbon of the first glucose and an oxygen molecule on that second glucose. And so you see over on the right-hand side of the diagram, we have joined these molecules. So two monosaccharides were joined together to form a disaccharide. This particular disaccharide is called maltose. We can break that apart. If we take maltose, we could split that with the hydrolysis reaction, where we break water, add water back across that bond, and we can break this down back into two glucose molecules. We can continue this condensation or dehydration synthesis reaction, and we can just keep adding more and more and more glucose onto this growing chain, and that would form something called a polysaccharide. A polysaccharide is a polymer, a chain of monosaccharides. Anytime you see the word polymer, a polymer just means it's a chain of repeating subunits, and in this case, that subunit is the monosaccharide. It's a sugar. So we're going to make a chain of sugar. And so you see here in this diagram, each of these green hexagons represents a glucose molecule. So we keep repeating that condensation reaction over and over and over, keep adding more and more glucose to the growing chain. We would call this a polysaccharide. Specifically, it's starch. This is the way that plants store excess sugar. They form the starch molecule. Animals store glucose a little bit differently. They form something called glycogen. Glycogen is also a chain of glucose, thousands of glucose molecules put together in a chain. The chain is branched a little bit differently, more branching in the glycogen than we saw in the starch a moment ago, but it's still just thousands of glucose molecules put together. Animals store glycogen primarily in the liver, but also in skeletal muscles. We can break these down. So if we needed to break up glycogen or break up starch, simply that hydrolysis reaction, and that's going to chop glucose off one at a time. There's one additional polysaccharide that we need to look at, and this is cellulose. Cellulose is a structural carbohydrate that's found in the cell wall of plants. That's what makes the cell wall of plants very rigid. Cellulose is a chain of glucose. Now, if you look at this closely and then compare this back to the starch, what you will notice is that the orientation of the bonds between the individual glucose molecules is different. Here in the cellulose, if you see that oxygen that's between the glucose molecules, you see that alternates, one's up, one down, one up, one down. Whereas in the starch or in the glycogen, they are all oriented on the same side. And so that makes a big difference because we do not have digestive enzymes that are required to break down the alternating bonds. So we can digest starch, we cannot digest cellulose, and it's due to the orientation of these bonds between the glucose molecules. So whenever we have cellulose in our diet, we call that roughage. That's fiber. That's non-digestible material.